Welcome to this module on synthetic biology. So, what is synthetic biology? No, clearly, most people have never heard of this in their life. Well, to use the dictionary definition,
an interdisciplinary field that combines biology, energy, and computer science to design new biological parts and devices, or redesign existing biological systems for useful industrial purposes.
Okay, but what does that mean? Well, synth bio covers a lot of material. It combines biology, chemistry, computer science, and of course, artistic skill to create something wholly unique. Using technology to change biology and make some product, be it medicine, food, or much more, it can be broken down into some basic processes that it shares with other fields. Identifying a problem that you want to solve. Researching the background, what it is being done, and what is the problem. Laying a plan of action, how you will solve this problem, what systems and skills you will pull together, and of course, testing, and more testing, iterations to your project until it is done. Let's talk about some history. It is important to know that the term synth bio is only about 30 years old, but it is built on centuries of progress, especially in the past 100 years. In the 1930s, several groups were working towards finding out how genetics and inheritance worked on a molecular level. At the time, there was debate over whether DNA or proteins carried genetic information. By the 1940s, a team of scientists led by Oswald Avery, Colin McLeod, and Macklin McCarty showed that DNA could be transferred between bacteria, allowing a harmless bacteria to become harmful. In 1952, DNA as the hereditary material further confirmed this by showing viruses called bacteriophages inject their DNA into a host bacteria, causing injection, but does not inject protein, meaning DNA makes proteins, making it the primary genetic material. But the actual structure of DNA was still unknown, which would need to be solved if you wanted to actually work with it and manipulate it. In 1958, the world would learn that DNA has a double helix structure, two strands wrapped around each other. This was solved by James Watson and Francis Crick, using data in the form of an image of the helix stolen from a chemist named Rosalind Franklin, who was left out of the reports and did not receive recognition for her contribution. These discoveries allowed scientists to piece together a map of how DNA goes on to command the body. This map is known as the central dogma, that DNA makes RNA makes protein, transcription and translation. But it isn't that simple. DNA makes DNA as well through DNA synthesis. RNA can make DNA under special circumstances through reverse transcription. There are several types of RNA used at every step, and not all of it codes for proteins. And proteins are used at every step to read the DNA, assemble the RNA strands, and make sure it moves smoothly so you don't get things like cancer. Let's take a closer look at DNA. It is a double helix of strands containing base pairs that is chock full of things like coding regions, regulators, and large amounts of quote-unquote junk DNA that codes for nothing really. But those regulators, who are where we want to look, they decide when, where, and how much DNA we're reading and what's being produced. These command posts were the center of research for the 50s and 60s, when in 1961, French scientists François Jacob and Jacques Monod discovered on one of the first and most widely distributed regulator, the lactoperon. Short for lactose operon, this little buddy has two states it can be in, open or closed. It is closed when glucose is present and open when glucose is not present, but lactose is. The operon is made of several regions. A promoter, that is the beginning section that includes proteins such as polymerase, which reads DNA to this gene. Followed by the operator that actually allows the DNA to be read, the structural sections where the actual DNA is read and undergoes transcription, and eventually translation into proteins. A repressor is present in the closed state. The repressor is stuck to the operator, blocking the path of the polymerase, preventing the reading of the structural section. If glucose is not present, then lactose can bind to the repressor and cause it to break away from the operator, freeing the polymerase to carry on its way. By the 70s, we're finally ready to modify these disparate pieces. Molecular cloning through the polymerase chain reaction, PCR, allowed the production of millions of pieces of DNA. Important as the gene is being studied is only a small fraction of the total found in the cell. By amplifying and elongating this single gene, work can actually be done in a cost and time-effective manner. As seen by the creation of synthetic human insulin in 1978, allowing insulin to be made inside of bacteria that can be purified rather than pulling it out of a pig or cow. By the 1990s, DNA could be sequenced using the quickly advancing computer technology, meaning the millions of DNA base pairs could be read in the time it would take to do a handful of them doing it by hand. By 1997, the University of Wisconsin, through its E. coli genome project, had completely sequenced the E. coli bacteria, the primary bacteria used in research. This gave a substantial boost for the Human Genome Project, which had been going on for several years at this point. But this still wasn't a quick project. The human genome has over 600 times the base pairs and over 25 times the genes of E. coli. It would be over a decade more and finished in 2004, unlocking the door for the easier study of human genetics. In 1999, Golden Rice was released. This special grain has been modified to include genes from carrots to produce extra vitamins for areas facing malnutrition. In 2000, the first synthetic regulator is produced. By 2003, standardized systems were created to allow easy, compatible research. Teams across the world could now use building blocks that could easily snap into place. In 2004, the first conference of the International Genetically Engineered, or iGEM, machine meets at MIT. If it feels like it's accelerating, it is. 2006 saw a bacteria destined to fight cancer. 2008 saw an entirely recreated bacteria genome made from scratch. 2012 saw CRISPR-Cas9 developed, allowing the widespread and simplified use of genetic engineering. In 2020, organic robots called xenobots were developed, making a jump into the synthesis of organic and mechanical. And in 2021, you probably recognize this one, the first human-approved mRNA vaccine is produced in under a year, a record-breaking achievement. But really, it's only the beginning, as new methods and techniques furthers the possibilities of the future. One such group pushing the envelope is iGEM. iGEM is an international association and competition for teams at the high school, undergrad, and above levels that aim to promote advancement of the synthetic biology field, focusing on the design, development, and integration of a project using synthetic biology is the name of the game, as is developing marketing material, presentations, and outreach ideas. With hundreds of teams across the world, many great things are possible. iGEM divides itself into villages. There are too many to get into them in any depth, but some would include diagnostics and therapeutics, manufacturing in space, agriculture and conservation, and many more, each suited for specific niches synth-bio and biotech are used in, allowing students like you or I to help shape the future. This was a presentation by the Penn State Synth-Bio Club and iGEM team.